Okay, then our topic is now the topic is digestion and absorption. Before the topic is called the anatomy of digestion, we will discuss. Now we will discuss what are the glands are associated inside and outside to support the digestion. Okay. Absorption. So, what are the glands are present inside or outside? It is to be supporting for digestion. Supporting for digestion. The first we have to see in our mouth or buccal cavity. Mouth or buccal cavity. Buccal cavity. So what are the glands are associated in buccal cavity to help digestion, So now we will see to discuss what are the glands are to be associated. Okay, here the 4 pairs of saliva glands is to be present, but human beings is having only 3 pairs of saliva glands. So another pair it is found in only animals, that is called infraorbital saliva glands. So it is absent for human beings, it is presenting for animals. So that 4 pairs of glands we will see where the locations and connect the different types of ducts. We will see now. Mouth salivary glands. Salivary glands. We will see now salivary glands. Okay, so now we will see the salivary glands. The first saliva gland, it is to be found in ear pinna. So the saliva gland is to be found in ear pinna. This gland is called parotid gland. Parotid saliva gland. Okay, so it is duct. It is to be releases in molar region. So the duct will be released in molar region. So this duct is called Stenson's duct. Okay, the duct name is called Stenson's duct. Another pair of saliva gland is to be found in lower jaw and upper jaw. It is found, both are combined there in this region is having another gland. So the gland is called submaxillary or submandible. So the gland name is called submaxillary or mandible. Okay, so this is having duct. This is also to be releasing premolar region. So the duct name is called Wharton's duct. The duct name is called Wharton's duct. And one more pair of saliva gland is there, that is below the tongue. Below the tongue. This is tongue. So below the tongue. This is also having duct. This is also having duct. Okay. So this gland is called sublingual. Sublingual. Sublingual gland. The duct is called ravenous duct. Ravenous duct. Okay, ravenous duct. Okay. Another fourth pair of gland is there. That is absent in human beings. It is to be present in animals. That gland is to be present in below the eye orbits. Below the eye orbits. Okay, below the eye orbits. It is absent for human beings. The gland name is called infraorbital. Infraorbital. Infraorbital saliva gland. Saliva gland. Okay, infraorbital saliva gland. So absent in humans. Absent in humans. Absent in humans. Okay, this gland is to be absent. This gland is to be absent. It is found in animals. It is found in animals. Okay, this is in mouth. Mouth leads saliva glands. Salivary glands. In these glands, located in different places. Located in different places. The parotid gland is to be located in ear pinna, and submaxillary or mandible gland is to be located in this region, in this neck region, neck started region. And one more gland is called sublingual. One more gland is called sublingual. This is below the tongue. Okay, the duct is having ravenous duct. The duct is having ravenous duct. So okay, this is the location of saliva glands. Location of saliva glands. So this is first pair. This is second pair. This is third pair. The fourth pair is absent in human beings. Fourth pair is absent in human beings. Fourth pair is absent in human beings. Human beings. Okay. Okay, here the salivary glands. Okay, the salivary glands, it is releasing a mixture of saliva. Mixture of saliva. Mixture of saliva. The saliva is releasing a mixture of saliva. Mixture of saliva. Mixture of saliva. Saliva releasing mixture of saliva amylase. Saliva amylase. It is called as ptyalin. It is called as ptyalin. It is called ptyalin. The saliva releases and mucin and lingual lipase also to be released. Lingual lipase enzyme. Lingual lipase enzyme. The saliva glands, okay, releases a mixture of saliva amylase enzyme. That enzyme is also called ptyalin. That enzyme also called ptyalin. And also saliva releases mucin and lingual lipase. The mucin and lingual lipase is small amount has to be releasing. So it is most of the times releasing in ptyalin amylase. It is called ptyalin enzyme. Ptyalin enzyme. In ptyalin enzyme going to work. Ptyalin enzyme going to work in carbohydrates. Carbohydrates. Means the ptyalin enzyme going to work carbohydrates converted into disaccharides. Okay. The ptyalin enzyme going to work in carbohydrate. Okay, the ptyalin enzyme will going to work to convert the carbohydrates. Going to work carbohydrates to convert disaccharides. Nearly 30% of starch will be digestion. Nearly 30% starch. Nearly 30% starch will be digestion will be here. 30% starch will be digestion will be there. Okay. The saliva gland releases the mixture of saliva amylase. It is also called ptyalin. The ptyalin is going to work carbohydrates. That carbohydrates are converted into disaccharides. Nearly 30% of starch will be digested in our mouth. That is called small sugars and small fats. And chlorine ions. Some mucin and lingual lipase. It is a small amount will be releasing in our saliva glands. Mucin means it is also to be releasing for mucus. Means it will be create a bolus. And whenever we crushing the food particles to mix mucus to create a bolus. Okay. Next, lingual lipase means it is a small fats. The lingual lipase is to be going to work on the small fats and fatty acids. Okay. This is the saliva glands. Next we will move to stomach. Next we will move to stomach. Okay. Next, now we will discussing stomach. Okay, this is stomach. This is called stomach. Okay, this is called cardiac region. This is called fundic region, and this is called pyloric region. Okay, in stomach is having some gastric glands have to be present in stomach. So the gastric glands have to be present in our stomach. Where it is to be found, the locations we will see and discuss clearly. Okay, the cardiac part is nothing is here. In fundic region is having three types of glands is to be found. First, it is to be found in oxyntic cells. Oxyntic cells or parietal cells. These are to release HCl, and one more thing is causes intrinsic factor. Intrinsic factor. Okay, the causal intrinsic factor, it is going to absorption in B12 vitamin. Okay, going to absorption B12 vitamin. B12 vitamin. It is called cyanocobalamin. It is called cyanocobalamin. Okay, the first gland is called axonic cells or parietal cells. They are secreted HCl and causal intrinsic factor. Causal intrinsic factor. HCl going to work to activate the enzymes. HCl is going to work activate the enzymes. So the first thing is the HCl it is to be working on first and stomach. The HCl to kill the bacteria. To kill bacteria. Kill bacteria. To kill the pathogens. Kill the pathogens means in stomach the HCl to kill bacteria. Kill bacteria or bacteria. Bacteria HCl to kill. HCl killed. HCl is to killed. HCl is killed. Okay. The first the bacteria will be going to die because of the HCl will be going to take action to kill that. Going to take action to kill that organisms. Okay. Next, one more thing is chief cells. Chief cells or peptic cells. Peptic cells. These cells are to be releasing two pro-enzymes. Two pro-enzymes. One is pepsinogen. Pepsinogen. Another thing is prorenin. Prorenin. These two are pro-enzymes. 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 These two are pro-enzymes. Okay. These two enzymes are inactivated stage. These two enzymes are inactivated stage. And the HCl is going to activate these two enzymes. Two enzymes means the pepsinogen is going to activate pepsin in presence of HCl. Okay, the HCl is converted into pepsinogen to pepsin and prorenin to renin. Prorenin to renin. We will see now. Okay, this is pro-enzymes and inactive enzymes. Next here mucus. It is to be secreting mucus. Secreting mucus. Secreting mucus. Secreting mucus. Okay, secreting mucus. Secreting mucus. Okay, this is called mucus cells. This is called mucus cells. Secreting mucus. Secreting mucus. Secreting mucus. Okay, right. Okay, here the HCl. How to activate? Pepsinogen is pepsin and prorenin is renin. These two are pro-enzymes. These two are pro-enzymes. Okay, the pepsinogen going to convert in HCl. Going to convert in HCl is pepsin. Is pepsin. Next, prorenin. Prorenin is going to convert HCl in renin. In pyloric part. In pyloric part is having G cells. Pyloric part is having G cells. Means gastric cells. Gastric cells. In these gastric cells to releases gastric hormone. Gastric hormone. Gastric hormone. Okay, this gastric hormone going to stimulate all gastric glands. All gastric glands. right? G cells, it is also called gastric cells. Okay, it is called releasing for gastric hormone. This gastric hormone will be stimulate all gastric glands. All gastric glands. Okay, this is a stomach region. Means four things are to be found in. One is oxalic cells or parietal cells. They are releasing HCl and causal intrinsic factor. HCl is activated to pro-enzymes. Okay, what are the pro-enzymes? One is pepsinogen, another one is prorenin. They are two are pro-enzymes. It is called inactive stage. It is releasing for inactive stage. The HCl will be going to work to convert then enzymes, then activated enzymes. At the same time, HCl is going to kill the pathogens in our stomach. Okay, next causal intrinsic factor to be absorption of B12 vitamin. Absorption of B12 vitamin, that the name is called cyanocobalamin. Next, chief cells are peptic cells. Chief cells are peptic cells. These cells are to be releasing pepsinogen, prorenin. These two are proenzymes, means it are inactive stage. Inactive stage. Whenever mix the HCl to convert activated enzymes. Next, mucus cells. These mucus cells are said to be secreting mucus. Mucus means why means secreting mucus? Why means it is may damage the walls in HCl, so that's why it will be secreting mucus to protect the stomach walls or intestinal walls or duodenum walls. Okay, next, this is pyloric region is having G cells. Pyloric region is having G cells. This G cells means gastric cells. Gastric cells are to be releasing for gastric hormone. That gastric hormone will be stimulate all gastric glands. Okay, this is the stomach. Next, we are coming to the first thing is liver. Next one is liver. Next, going to liver. Okay, before the liver, intestinal glands is there. The intestinal glands are to be found in. So one is the duodenum part. Okay, another one is the small intestine. We will discuss this intestinal gland. First, we will discuss in liver. Right, this is liver. Okay, liver, and another one is pancreas. Right. Okay, this is liver. This in liver is having so many lobules. It is called so many lobules. This is called lobules, and then lobules is connected in hepatic duct. Hepatic ductules. The ductules are to be connected into the hepatic duct. Okay, these all are lobules. That lobules are to be connected into the ductules. That is called ductules. That all ductules will be connected into one duct. This is called hepatic duct. Hepatic duct. Hepatic duct. Next, this duct in this region only. In this region is having bile duct. Bile duct. Okay. This is called gallbladder. This is called gallbladder. Okay. In this duct it is called cystic duct. Cystic duct. Okay. And this duct is called common bile duct. Common bile duct. Okay. Common bile duct. Okay. This is called pancreatic duct. And this is called hepatopancreatic duct. Hepatopancreatic duct. Okay. This is called hepatic bile duct. Hepatic bile duct. Okay. This sphincter already we will discuss. The sphincter is called OD sphincter. Okay. Now we will see the liver. The transverse section of liver. So we have to cut a piece. Cut the piece. We will see the transverse section of the liver. We will see the liver. Okay. Right. Okay, this is liver. Okay, it is having many lobules. This duct will be dispensed in different types of duct. Hepatic ductules. This is called many lobules. Many lobules have to be connected in hepatic ductules. All hepatic ductules will be connected in hepatic duct. Okay, this is called gallbladder. And this is called cystic duct. Okay, cystic duct. And this is called bile duct. This is called common bile duct. And this is also, this is pancreatic duct and hepatopancreatic ducts. So we will see the transverse section of the liver. In diagrammatically. We have to cut one piece. We have to cut the transverse section of the liver. transverse section of the liver. Centrally placed on central vein or radial vein. Central vein or radial vein. It is to be releasing for a ring. It is to be releasing for a ring. It is like a ring. This is called ring. So this is hepatic cord. This totally the ring is called hepatic cord. Hepatic cord. This ring is called hepatic cord. Hepatic cord. Hepatic cord. Okay. These cells are called hepatocytes. Hepatocytes. The cells are called hepatocytes. Hepatocytes and centrally placed. This is called central vein or radial vein. Next, this in middle of this place is called sinusoids. Sinusoids. Okay, this is called hepatic cord. These cells are called hepatocytes, and this is called central vein. This is called central vein, and this in border means in border is having Gleason's capsule is there. In the membrane is having, so here it is to be found in Gleason's capsule. Gleason's capsule. Okay, the hepatic means it is called hepatic cord. These cells are hepatocytes. The in middle empty place is called sinusoids. In the membrane, the wall is having, it is called Gleason's capsule. Right. We will see here many lobules. Okay. The structural and functional unit of liver is called lobule. The structural functional unit of liver is called lobules. Is called lobule. Lobule or lobules means it is structural and functional unit of liver. Here many lobules. The many lobules are to be connected. The ductules, hepatic ductules means the hepatic ductules are to be collected bile juice. Who is to release the bile juice? The hepatocytes. This is called hepatocyte. The cells is called hepatocytes. These hepatocytes are to be releasing bile juice. In this bile juice to collected hepatic ductules. Then collected hepatic ductules to be going to hepatic duct. Hepatic duct to common bile duct to gallbladder. Gallbladder is having cystic duct. After it is called common bile duct to it going to pancreatic duct to then duodenum part. So the hepatocytes are the bile juice producers. Okay, hepatocytes are bile juice producers, and the middle empty place is called sinusoids, and the membrane or cell wall is having Gleason's capsule. Right. Okay. This is a liver structure. This is the liver internal structure. So what are the functions doing the liver? What are the functions are doing liver? We will see now. We will see now. Okay. This is location of liver, location of pancreas. We will have to discuss the pancreas after the liver. We will discuss pancreas. Okay. Okay, this is called liver. The liver is having many lobules. The many lobules are to be connected in hepatic ductules. That hepatic ductules are to be connected hepatic duct to common bile duct to cystic duct to gallbladder, and common bile duct to hepatopancreatic duct, and hepatopancreatic duct to duodenum. Then juice will be released into the duodenum part. Okay, in this internal structure is called the liver curd. This is central vein to releases hepatic curd. These cells are hepatocytes. In the middle place is called sinusoids. In the walls is having some capsule and jelly-like material that is called Gleason's capsule. Next we will see what are the functions are doing liver. Okay, we will see what are the functions. We will see what are the functions of liver. Functions of liver. First, the liver will be secrete bile juice. The first thing is bile juice. Bile juice contain no enzymes. Contain no enzymes. This bile juice was preparing for fat digestion. The bile juice will be, the liver is secret bile juice. That bile juice containing no enzymes. There is no enzymes is to be present in bile juice. So the bile juice, it is to be preparing for fat digestion. Preparing for fat digestion. The process is called emulsification. The process is called emulsification. Emulsification means, okay, that fat will be break down into small molecules to coated bile juice. Coated the fats in bile juice. Okay. The bile juice is to be preparing for fat digestion. That process is called emulsification. Means the fats will be, large fats will be break small particles to coated bile juice. After then lipase enzyme will be working. After coated fats, lipase enzyme is working. Working to break down the parts. Break down that fats. Right. Next, second function is called carbohydrate metabolism. Carbohydrate metabolism. The carbohydrate glucose. The carbohydrate end product is called glucose. It is glucose. Okay, glucose is normally coming from carbohydrates. Okay, the carbohydrate metabolism is three things. Okay, the three things is called, the first one is glycogenesis. And the second thing is, okay, glycogenolysis. Okay, the first thing is glycogenesis, second thing is glycogenolysis, and third thing is gluconeogenesis. Okay. The carbohydrate metabolism is three things. First thing is glucogenesis. Glucogenesis means the formation of glucose. Where it is from? Glycogen. From glycogen to conversion of glucose. Glycogenesis means the conversion of glucose. It is called glycogen. Okay, conversion of glucose. And glycogenolysis also conversion of glucose. Means using glycogen. The glycogen will be converted into glucose. That process is called glycogenolysis. And next, glycogenesis also, glycogen will be converted in glucose. These two are conversion of glucose. Then finally gluconeogenesis. It is a gluconeogenesis, a conversion of glycogen. Glycogen means it is glucose will be converted. Glucose will be converted glycogen. These two are conversion of glucose. Gluconeogenesis is called a conversion of glycogen. Means the glucose will be converted into glycogen. The glucose is converted into glycogen. Okay, Okay, this gluconeogenesis means it is a small fatty acids and amino acids. Okay, and proteins. Okay, and glucose proteins. Okay, they are different. Okay, it will be conversion in the gluconeogenesis. Only not one glucose. It is a conversion of what is that? Fatty acids and amino acids. Okay, and proteins or pyruvic acid. It is to be a conversion of glycogen. It is a preparing of glycogen. Okay, this is first thing is bile juice. Okay, the working of bile juice means bile juice is not containing enzymes. It is to be going to work preparation of fats digestion. That process is called emulsification. Emulsification means it is a coated. It is called coated fats in bile juice. The bile juice will be coated into fats. Okay, then the lipase enzyme will be working. Next, carbohydrate metabolism. Carbohydrate metabolism having three things. One is glycogenesis. Another thing is glycogenolysis. Third thing is gluconeogenesis, means the glucose is conversion into glycogen, remaining two are the glycogen will be converted into glucose. Okay, in gluconeogenesis process is having some fatty acids and amino acids and pyruvic acids, they are also converted into glycogen. Okay, next thing is detoxification. Next function is called detoxification. Next function is called detoxification. Detoxification means, for example, in stomach, we will eat different types of food. And drink alcohol, and different types of drugs will be injected. Okay, so this all poisonous food, and we have, for example, we will take in any poisons and any alcohol. Okay, there is a highly toxins. It is called harmful toxins. Okay, that all heavy toxins or harmful toxins or poisons, whatever it is, they are directly going into the liver. This is called hepatic portal vein is there. So everywhere hepatic portal veins will be connected into the directly liver. This is called hepatic portal veins. The hepatic portal veins is everywhere is connected to collect harmful toxins. That all harmful toxins will be going to enter the liver. That liver will be converted into detoxification. Detoxification means the highly harmful toxins will be reduced. Highly harmful toxins will be reducing. That is called detoxification. Detoxification means, so whatever it is, we will eat in some poisons and alcohol. Okay, that's time that all poisonous food or all poisons and toxins, they are connected hepatic portals, that going to directly through hepatic portal veins to liver. The liver is converted in detoxification. Detoxification. It is small amount, not is a large amount. Okay, next function is called ammonia is converted into urea. Next function is called ammonia is converted into urea. Ammonia is converted into urea using ornithine cycle. We will discuss clearly in excretory system. Ornithine, what is ornithine cycle? How to convert ammonia into urea. Okay, we will discuss in clearly in excretory system. Okay, so it is one of the function. Next one is called lipid metabolism. Lipid metabolism means excess amount of fat. Okay, the lipid metabolism means the excess amount of fat will be stored in liver. In the form of triglycerides. Understand? Okay, it is called lipid metabolism. Lipid metabolism means excess amount of fat will be stored in liver in the form of triglycerides. Next Next function is called producing heat. Producing heat. Producing heat. And another function is called producing blood clotting agent. Produce blood clotting agent. Blood clotting agent. Blood clotting agent means it is to be releasing, what is that? Fibrinogen. It is releases fibrinogen and prothrombin. Prothrombin and anticoagulant heparin. Anticoagulant agent is called heparin. Okay, blood clotting agents also to be releasing. Blood clotting agents also to be producing liver. And one more function is called Cori cycle. Cori cycle means it is a conversion of lactic acid. Means, okay, the Cori cycle we will discuss clearly in respiratory system. Okay, for example, so we will walk or exercise. Okay, that all organs will be coordinated. So all organs is having depth oxygen molecules. So the breathing will be fast. And oxygen will be depth. That's the time the liver will be releases lactic acid, and lactic acid will be conversion of pyruvate, and pyruvate to phosphates, adenosine phosphates or ATPs. Okay, one ATP is called one glycerides. What is this? One lactic acid is conversion of glycogen. That glycogen is called, it's a 36 ATPs or 32 ATPs. It will be releasing. Okay. Next, we will discuss clearly in respiratory system. What is the process of Cori cycle? Okay. Next coming to the pancreas. Next, pancreas. Next is called pancreas. Okay, the pancreas is to be acting endocrine and exocrine. This is also called mixed gland. The endocrine will be releases insulin and glucagon, and exocrine will be releases pancreatic juice. We will see the pancreatic transverse section. We will cut the piece. We will see the pancreatic transverse section. This cells is called acinar cells. Acinar cells. Acinar cells. These cells are islets of Langerhans. Islets of Langerhans. Islets of Langerhans. It is to be acting endocrine. It is to be acting endocrine gland. Endocrine gland. Endocrine gland. It is to be acting exocrine gland. It is to be acting exocrine gland. Okay, these acinar cells are to be releasing pancreatic juice. That pancreatic juice is containing enzymes. Containing enzymes. What are the enzymes will be to releasing acinar cells? So, how many enzymes are to be releasing acinar cells? Okay, we will see. Okay, this is a transfer section of the pancreas. Transfer section of the pancreas. Okay, such amount of cells, maximum amount of cells will be acinar cells. Remaining it is to be found in islets of Langerhans. This is acting for endocrine. This is acting for exocrine. This is also to be releasing for enzymes or pancreatic juice or pancreatic juice or enzymes. Okay, that juice is having enzymes. That juice is having enzymes. So, what is the enzymes will be present in the pancreas? Okay, first it is called trypsinogen. Trypsinogen. First enzyme it is called trypsinogen. Second one is chymotrypsin. Chymotrypsin. Okay, chymotrypsin. Chymotrypsin. First one is trypsin, and second one is chymotrypsin, and third one is carboxypeptidase. 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 And third one is fourth one is nuclease. Nuclease. And fifth one is having, what is this? Fifth one is having, what is that fifth one? Amylase. Amylase. And sixth one is having lipase. Sixth one is lipase. Lipase. This is also called triopsin. This is also called triopsin. This is also called triopsin. Another name is called triopsin. Another name is called triopsin. Okay, the pancreas is acting endocrine and exocrine. Okay, the endocrine will be releases insulin and glucagon. Exocrine will be releases pancreatic juice. We will see the transfer section of the pancreas. The most of the cells are to be present in acinar cells. This acinar cells is to be releasing for pancreatic juice. That pancreatic juice is containing enzymes. That pancreatic juice is containing enzymes. Trypsinogen, chymotrypsin, and carboxypeptidase, nuclease, amylase, and lipase. That lipase, another name is called triopsin. The most of the times is asking triopsin, not lipase. Not lipase. Okay, this is pancreas. This is pancreas. Now we are moving intestinal glands. Intestinal glands. Intestinal glands. Intestinal glands. Okay, the intestinal glands means so one is duodenum, and jejunum, ileum, and ileum. Okay, The duodenum is having submucosa layer, and this is also having 5 layers, but this is ileum and jejunum is having only mucosa layer. Mucosa layers. It is having Brunner glands. And mucosa here is having crypts of Leverkusen. Crypts of Leverkusen. Okay. These two are intestinal glands. These two intestinal glands is to be releasing succus enterica. Succus enterica. Also releasing for enzyme activator. Also it is releasing for enzyme activator, that is called enterokinase. Enzyme activator also releasing. It is called enterokinase. Enterokinase means, for example, trypsinogen. Will be activate the enterokinase, enterokinase. It is called trypsin. Okay. Also it will be releasing enzyme activator and enzymes. So what are the enzymes will be released? Okay. The enzymes. How many enzymes are releasing? 6 enzymes also to be releasing. So what is the enzymes? The first thing is called tripeptidase, and the second thing is called dipeptidase, and the third thing is called aminopeptidase, and fourth one is nucleodase, and nucleosidase, and last thing is lipase, Okay. The first one is tripeptidase. Tripeptidase. And second thing is dipeptidase. Next, third thing is what is that? Third thing is aminopeptidase. And fourth one is what is? Nucleodase. And the fifth one is having nucleosidase. Sixth one is having lipase. Okay. Duodenum, jejunum, ileum, intestinal glands. In 3 parts it is to be found in 2 intestinal glands. One is Brunner glands, and second one is crypts of Leverkusen. These two are releasing succus enterica. Succus enterica means that is also, it is to be activating enzyme activator, that is called enterokinase. Trypsinogen will be activated into trypsin. Okay, this trypsin only working in alkaline medium. Highly acidic medium, the enzyme is working is called pepsin. Okay, highly acidic medium, the enzymes are working. They are working in stomach, pepsin. Okay, in intestinal it is working, it is called trypsin. Okay, they are replacing in stomach and stomach is replacing intestine, so they are not working. Okay, so trypsinogen is activated in enterokinase into trypsin, and also to be releases enzymes. So this is tripeptidase, dipeptidase, aminopeptidase, and nucleodase, and nucleosidase, and lipase. These enzymes are to be releasing in intestinal glands. Okay, so our topic is completed. The first it is called in buccal cavity. Buccal cavity means mouth. Mouth is having saliva glands. 3 pairs of saliva glands is present in human beings, and fourth infraorbiter is to be absent in human beings. Okay, the saliva it is releasing for saliva amylase. It is also called thylacine. And next comes to stomach. In fundic region is having 3 types of glands is there. One is oxyntic cells or peptic cells. They are releasing HCl and causes intrinsic factor. HCl kill 2 pathogens in our stomach, and causes intrinsic factor to absorption of B12 vitamin. Next, one more thing is called chief cells are peptic cells. That cells are to be releasing 2 pro-enzymes. That 2 are inactive stage. To activate the HCl. The 2 enzymes are, one is pepsinogen, another one is proninin. Next, another one is called mucus cells. Mucus cells is to be secreting mucus. Next, coming to pyloric region. G cells are to be present in the pyloric region. G cells means gastric cells. Gastric cells are to be releasing gastric hormone. That gastric hormone is stimulate all gastric glands in our stomach. Next, coming to the liver. Okay, the liver is having many lobules. That lobules is to be connected in hepatic ductules. That hepatic ductules is to be connected in hepatic duct to bile duct to connected cystic duct to gallbladder. Next, common bile duct to connected directly in where? Hepatopancreatic duct. They are going to releasing in duodenum. The juice will be releasing duodenum. The transfer section also we will see. Okay, the many lobules are there. Hepatic cord also there. Hepatocytes also there. Okay, the hepatocytes also to be released bile juice. Next, the liver is doing many functions. We will discuss. The first thing is bile juice not containing enzymes because of preparing for fat digestion. Fat digestion means also called emulsification. That process is called emulsification. Emulsification means it is a coated fats, means the bile juice will be coating on fats. Then finally lipase enzyme will be work. Next, second one is called carbohydrate metabolism. The carbohydrate metabolism is having 3 things. One is glycogenesis, second thing is glycogenolysis, and third thing is gluconeogenesis. First 2 things is called formation of glucose in the form of glycogen, and third thing is called glyconeogenesis. Glyconeogenesis is called the formation of glycogen from the form of glucose. Next, third function is called detoxification. Only mean we will drinking water and food. Okay, we will take the poisons and different types of poisons that fall highly toxic substances. That all toxic substances will be go through the hepatic portal veins to go to liver. That liver is to be detoxificated. That liver is to be detoxification. Next, ammonia will be converted into urea. Ammonia will be converted into urea using ornithine cycle. Using ornithine cycle, and also producing heat. And also to be producing heat. And next, producing blood clotting agents. And also to be producing blood clotting agents. What is that blood clotting agents? Prothrombin and fibrinogen and anticoagulant heparin. And one more function is called Cori cycle. The Cori cycle is called the lactic acid. The lactic acid is converted into the glucose. Lactic acid is converted into the glucose to give an energy. And one more thing is called lipid metabolism. Lipid metabolism means the excess amount of fat will be stored in liver in the form of triglycerides. Okay, so the many functions are doing liver. Next, coming to pancreas. The pancreas is acting for endocrine and exocrine. Okay, so we will discuss only exocrine. Okay, we will see the transfer section of pancreas. The acinar cells are to be present. That acinar cells is to be releasing pancreatic juice. That pancreatic juice is containing many enzymes. Next, coming to the intestinal juice. Intestinal glands. Intestinal glands means, so the one gland is found in duodenum part and submucosa, that is called Brenner glands, and jejunum and ileum is having mucosa layer. That mucosa layer is having crypts of liver cone. The two glands are to be releasing for succus centrica. It's nothing but intestinal juice. It is to be enzyme activator, that is called enterokinase. Enterokinase will be activating trypsinogen to trypsin, and also to be releasing enzymes. That enzymes is called this all. Okay, this topic is the glands, not a topic. The glands is over. Tomorrow we will discuss the physiology of absorption. Thank you.